This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 201, recorded on September 28th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from verticalfarm.com, Dickson Despommier.
Hello, Vincent.
Is that a good attribution for you?
Fantastic. I love it.
How you been?
I've been well.
I haven't seen you in days.
I've been traveling.
Good. Well, welcome back and welcome to TWiV.
Thank you.
We're entering the second 200 set of TWiV.
Wow.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
And I got it right this time.
Yes, I am, in fact, in Western Massachusetts this time.
Couldn't get it at the BSO4. I couldn't remember where he was from.
Well, I was sitting right there.
Right.
Yes, so we were informed.
Yes.
But it was just a hologram.
That's right.
Wouldn't
I beamed in.
Wouldn't that be nice? We could do this by hologram.
Yeah. Be like being there.
One day.
Exactly.
Also joining us today from North Central Florida, Rich Condit.
Hi, everybody. How you doing?
Hello, Rich.
How's everything, Rich?
Good. I'm just trying to figure out what the weather is in Celsius here.
I thought you were in Florida.
I am.
They're right. That's all right. We're up here in Fahrenheit.
87 Fahrenheit, 30.55 Celsius.
Nice.
Mostly cloudy.
Nice weather.
Well, half cloudy.
I thought you said summer was ending.
Well, I was wrong.
Because here it's 19, 19 and cloudy and rainy.
We had torrential rain.
Probably the same for you, Alan, right?
It's 12 Celsius.
Whoa!
54 Fahrenheit and pouring rain.
Wow,
No
Western
kidding.
I'll be
Massachusetts,
darned.
yeah.
That's miserable.
Yep.
Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everyone.
Hello, Kathy.
Hey, Kathy.
Hi, Kathy.
It is 66 Fahrenheit, 19 Celsius, and it's sunny with fluffy clouds. Just about perfect.
That's pretty cool.
You know, we
Yeah.
have the same temperature, but totally different. It's all cloudy here.
Well,
Yeah.
so I can see some blue out the window here, Vince. I think it's going to clear up later.
Well,
Ever
we
the optimist.
certainly
Yeah.
got enough rain.
We're the only ones that care about any of this, by the way. We just.
Oh, no. Everybody is absolutely fascinated with the weather on
Oh,
Twitter.
absolutely.
Well, when we go somewhere and we forget to do the weather,
Well,
I was in Dublin doing TWiV and, in front of an audience, and we started on the show and someone yelled out, "Hey, what about the weather?" That was great. Really. All right. This is sort of a post-apocalyptic show.
Yes.
We had a great 200, and I don't know how we can do the same, but we'll try. That was
How
just
long
so,
did it take to do 200 episodes?
It's been 4 years now.
About as long as it took to film episode 200.
That was a long film. When that film comes out, we'll talk about it. So it's actually 4 years ago this month, Dickson, that you and I started TWiV.
yeah, I was mostly filled with remorse and regret for not being able to make the 200th anniversary of this show. I was interviewing medical students, or potential medical students for this medical school, and I had that commitment already ironclad, and I couldn't get out of it, so I'm sorry.
We should have had one of those, cardboard photograph cutouts.
We usually use Kermit the Frog for all the students that don't show up for their photographs. I would have been a good Kermit.
This episode of This Week in Virology is sponsored in part by ASM, the American Society for Microbiology, the world's largest membership society for microbiologists. Find out how ASM membership can help advance your science, your career, and your network. Go to asm.org/advance. Today is an email episode.
Yes.
We have lots queued up, so let's get right started with the
We're
first.
just mailing it in today.
Yes, I have to read
love
this one. I have to read this one
episodes.
because
These are so much
because this one has a regret. Let me just take the short ones, but this is the one that I would voice my regret for too.
Okay, sure, it makes sense.
So it
Go
says,
ahead.
"Dear Vincent, it's to you. I guess maybe you should read this.
It's all right.
I am glad you had a chance to get a glimpse of the BSL-4 world. You might remember that I have been trying for 1.5 years to get you to see the Galveston National Laboratory, a facility with an actual active, that's in capital letters, program, and a number of very experienced PIs that currently work in the BSL-4. I wish we would have had the chance first, especially for TWiV 200, a very disappointed Dennis.
I got this,
Dennis,
I got this right after the episode was released.
Dennis, I have regrets, but my regret is that I couldn't have even been at the one up in Boston.
So Dennis has been, in fact, trying to get us, Rich and I, to go to Galveston for some time.
Right.
And we just haven't been able to do it. But we're sorry about that, right, Rich?
I'm sorry about it, yeah. We're sorry about it.
But I have a sense that in Galveston we couldn't actually get in the BSL-4, right?
Right.
Aha.
So, because it's active, whereas in Boston it was perfect. We got it not only in to do TWiV, but we got a tour which is filmed and will come out soon. So,
Yeah, we and soon the TWiV audience are the only people who have benefited from this long wait that they've had.
so I'm sorry, Dennis. Maybe 300 or 400 or 500.
250?
250. Do I hear
Whatever.
225?
Yeah, 225. I'm ready. My bags are packed. I'm ready to go.
Look, I'll clear my schedule for this one. I promise I will.
What, for
Well,
300?
how far
For
is
whatever the
Galveston
next one
from,
is.
How far is Galveston from Buda? It's probably pretty far, right?
Well, we did that calculation, actually. As I recall, it's maybe like a 4-hour drive or something. I may have to do it again. But yes, Buda being the center of the universe, that's an important calculation.
Yes.
I'll do it again while on the sly here. I'll see if I can multitask that one.
Well, you're going to do the next email, Rich.
Oh, I am? Okay.
Yeah, because you have some additional
Oh, yeah.
information, right?
Yeah, I do.
So you can't multitask right now.
Okay, fine. I'll save it. You want me to do this now?
Yeah, go ahead.
Okay. Jim writes. This is our friend Jim from Smithfield. Gents, a recent BBC health check program included a segment about rabies, which notes that vaccines derived from dog rabies may only be effective for people bitten by a rabid dog, but not a bat. I've attached the program, and it's the first segment, which is only about 4 minutes long. Dr. Behrens, linked to some of his advice from the London School of Tropical Medicine and Hygiene, is the speaker and source of the comment. Are there multiple vaccines for rabies? This complicates things.
I'll say.
Regards, Jim. So let's see. I actually emailed around when I got this to see if I could get some input on this, and I wound up contacting Matthias Schnell, who is at the he's a professor in the Department of Microbiology and Immunology and the director of the Jefferson Vaccine Center at Jefferson Medical College. And he wrote back and said, he sent me some PowerPoint slides from a lecture on rabies that he gives. And he says there are two things that are important to know. One, when people talk about rabies, they normally mean classical rabies virus. And that's RABV, R-A-B-V. And this is still the most important member of the genus Lyssavirus. Almost all deaths in human are caused by RABV transmitted by dogs. This is a big problem in developing countries, South America, India, Africa, China, Asia in general. In Europe and the USA, we only have rare rabies transmitted by domestic animals because we vaccinate, and that problem is gone. We still have a large rabies problem in wild animals, that is in the U.S. Skunks, raccoons, foxes. But if somebody gets bitten by a rabid animal, we do post-exposure treatment, which is 100% efficient if applied correctly. And I can comment on that later. About 30,000 people do receive post-exposure treatment in the U.S. every year. That's not cheap. We do have about 3 cases of rabies in the U.S. every year, but they were imported, people who traveled and got infected, or they got infected by bats. This brings us to the bat topic. All bat-derived rabies in the U.S. is classical RABV. So that's the classical rabies virus. So post-exposure will work. So the vaccine against rabies will work for bat rabies in the U.S. However, people might not realize that they've been bitten by bats, small teeth. So the problem in the U.S. is that more people do not realize they were exposed or do not know that bats are transmitting rabies virus. Second point, bats are very likely the natural host of lyssaviruses. Bats seem not to be as sensitive to rabies virus, and a lot actually have antibodies. That doesn't mean that they don't get rabies, but it is probably not 100% lethal. Makes sense if you think how close they live together. Also important, there are bat-derived lyssaviruses, which are antigenically different from classical rabies, RABV, so the vaccine doesn't protect against those. This is the case for the African lyssaviruses Mokola and Logos bat. So for these viruses, there is no vaccine, even though it would be easy to make one. Hope that helps. And he refers to his PowerPoint file. One of the things that I actually put here in the Google Docs thing for us to see is his phylogeny of the rabies viruses. And it can be broken down into two major phylogroups, phylogroup 1 and phylogroup 2, and the vaccine works on phylogroup 1 but not phylogroup 2. So it sounds like, yes, there is a bat rabies for which the vaccine doesn't work. It's not a problem in North America, or I guess in the West, but it might be a problem elsewhere.
Have there been human cases of this?
That I do
Africa?
not know.
Okay.
That I do not know.
What about the vampire bat transmission in South America? I know there have been some cases there with mortalities.
And I don't know whether that's the classic rabies virus or one of these others. I assume that it's the classic rabies virus.
Mm-hmm.
Okay. Mokola virus has reportedly caused 2 human deaths in Nigeria.
Okay. All right.
So it has happened.
So
But not enough to make a vaccine, I guess.
Right. Or not enough. We don't know of enough cases to
Yeah.
justify the vaccine.
So as long as we're on the topic of rabies, and I think this is there's somehow this is interesting because these emails all kind of fell into this session. There's a whole bunch of rabies going on, and it's ironic because I will skip ahead to my pick. Today, the day we are recording, September 28th, is World Rabies Day.
I'll be darned.
I'll be darned. What an irony. So I'll get to that later when we get to picks. At any rate, just a couple of more things since we're talking about rabies. This is a zoonosis. The virus is harbored in animals and transmitted to humans. It's not transmitted from human to human. It's transmitted by animals biting each other or other animals and humans. Where there are good vaccination programs for domesticated animals, it hangs out in the wild. So that's the case in the developed world and in the U.S. So the reservoir in the U.S. is skunks, raccoons, foxes, bats, etc. Most of the transmission in the U.S. to humans, apparently, if I understand it correctly, is from bats.
I think wild animal bites are the common transmission in the developed world.
Yes. Okay.
So bats would be in that group, but skunks
bats would be in that group.
Such.
Yes.
Yeah.
And in the developing world, where there's not good animal control, domestic animal control, and there's a lot of stray dogs who are unvaccinated. The dogs are the major reservoir and transmission vector to humans. Right. And the one last thing is that this is interesting because you get bitten, you get some local replication in the muscle cells, and then it infects neurons and travels up the axons of neurons to your brain, where ultimately it causes the symptoms. But that transmission can take a very long time. It usually takes a fairly long time, at least weeks, though if you get bitten in the head, it can take a shorter time. But it can take up to a year or maybe even longer, and that gives you time to actually apply a vaccine post-exposure, and that's an unusual situation. And so there is a vaccine, and if you're exposed to the virus and you get bitten by an animal and you strongly suspect or can prove that that animal was rabid, you can take the vaccine after the exposure, and that will prevent the disease, which is important because once you come up with neurologic symptoms, you're pretty much cooked.
It's too late. Yeah. You could also point out that the vaccine is no longer the really painful injections into the stomach. They're just in the arm. And I think if there's any suspicion at all of a bat bite, for example, you should go through that prophylactically, the vaccination.
As he points out, in the USA, there's 30,000 people a year who get this treatment. Because the risk of not getting there's no big downside, and the risk of not doing it is death.
One of the grad students in our department was coming home from a football game last year, I think it was last year, and kind of got brushed by a bat, and so he went through the vaccine treatment.
So I have two questions. One is that why did that other person refer to us as gents, because we're more than gents now? And Kathy, you should have spoken up.
It's an old email. It was before Kathy joined.
Okay, forgiven. Number two, Rich, how did you know today was National Rabies Day? And three,
you had two questions.
Okay. This is a part two of the second question.
This is his third of two questions.
And with so many viral infections in humans, does every day of the year have a virus associated with it?
Oh, that's a really good question. I have only 2 dates on my calendar. I have smallpox vaccination day, and I have World Rabies Day. And it's on my calendar. I don't know if it's actually repeating, and I don't know who brought that to my attention. But when something like that comes to my attention, I stick it on my calendar here on my iPhone, so I'm sure not to forget it.
I just found World Polio Day.
See?
Oh, is that right?
It's
what day is that?
October 24th.
It's coming up.
Great. I'll put it right down.
If there's no virus day for a lot of other days, perhaps we should have them. We should make them up.
Sure. Why not? You got to have an organization, though.
Pick 365 viruses that you'd like days for.
Rank them in order of importance.
So that's on my calendar. Today is World Rabies Day.
That's amazing.
When is Pox Day?
Smallpox vaccination day. I'm going to have to multitask that one for you.
All right, let me know.
I forget what else.
It's 2 hours. It's 2 hours. Or 3 hours 59 minutes from Buda to Galveston.
How long?
3 hours 59 minutes.
Now we're
about 4 hours. Okay.
We're now radiating in all directions.
So, because of this bat reservoir of RAB-V, it's not eradicable.
Right.
Yes.
Sure.
That's right.
Now, it just so happens that this week, in fact, yesterday, a paper was published in PLOS Pathogens in which is reported a novel rhabdovirus isolated from 3 individuals in Africa in 2009, with a hemorrhagic fever disease, such as that Ebola might cause in you. So this is a highly diverged rhabdovirus, which they're calling Bas Congo virus, BASV, but it causes hemorrhagic fever. So it's the first time that a Rhabdoviridae, a member of that family, has been associated with hemorrhagic fever. Dickson, do you know the 4 viral families associated with hemorrhagic fever?
I really should know them. I've tried to recite them daily, but I just have this mental block on them. But perhaps you could repeat them for the benefit of the audience.
It's not repeating. I haven't said them yet. Well, one is the filoviridae, right? Ebola, etc.
Sure.
Bunyaviridae, arenaviridae,
of course,
and flaviviridae.
Of course.
Does West Nile cause hemorrhagic fever, Dickson?
No, it does not.
Good, very good. You're awake. Anyway, that's a cool paper.
Smallpox vaccination day is May 14th.
Thank you.
Wow.
You may not be surprised to know that there is evidently no World Adenovirus Day. But
why is that?
I'm outraged.
I will get together with my colleagues and we will figure one out.
Is there a measles? There must be a measles day, right?
Sure.
I don't know.
It's a big one.
It should be easy to spot.
And a mumps.
Oh!
I think mumps comes on Fat Tuesday.
Alan, you're so quick. Holy cow.
Holy cow? No, that was for smallpox.
Yes.
Alan, can you do the next email?
Sure. Hi, TWiV crew. The Texas Wild Child was mentioned on TWiV 192. I'm listening to it now. Did you see the August article in Wired? Now, the Texas Wild Child was this rabies case, apparent rabies survivor who did not receive treatment and left medical attention. And is possibly out there walking around, having been a documented survivor of rabies.
Dropped off the radar completly.
Dropped off the radar. Teenager, right? Turned 18. So she just decided she was going to wander off, but she had shown up in an emergency room with symptoms of rabies. That's the point at which you're pretty much unrecoverable. She, was symptomatic neurologically. Recovered enough to leave the hospital and hasn't been heard from, but there's this possibility that somebody survived this virus on their own. And this Wired article is about rabies therapies that are being developed. And this is, I think we've talked about this before, haven't we?
We have the Wisconsin protocol. Is
the
that
Wisconsin protocol, putting somebody in a coma, and it's a series of
hypothermia.
Yeah,
I don't know which TWiV it was.
Yeah, there was one from Ohio like that too, wasn't there?
Right. There've now been several cases that have been treated with this protocol, and a number of them have survived. But as I think the article goes into, that this is still kind of really, really experimental.
Sure.
And nobody's quite sure which parts of it are working. I think part of it was didn't they give antivirals of some kind?
In which the Wisconsin?
Yeah, I think there was some ribavirin involved or something like that. And so it's a multivariable thing. We're not quite sure what worked.
It's not clear whether those would have any effect against a rhabdovirus, but of course everybody is terrified to remove anything from this protocol because it seems to have worked. An otherwise 100% fatal condition.
Yeah, I can't find the TWiV we did. We did discuss it. And then on TWiV 18, there's a letter from an individual about the Wisconsin protocol, and we respond as well. But yeah, we've talked about it before. It's something developed empirically. We don't really know why it works.
So before we go off the topic, I know that there's a few cases of spelunkers that got rabies just by inhaling. Does anybody have a record of that?
Actually, I've looked into that,
Rich
because that came up before, too, on some of our previous rabies things.
And
according to my contacts at the CDC who deal with this, they are more than suspicious. They basically don't believe that you can catch rabies by inhalation.
Okay.
So my guess is that there's no question, but that spelunkers in caves that are loaded with bats got rabies. But it's also now I'm making this part up, but it's pretty clear from these sort of brief encounters with bats where you don't even know that you got bitten, that probably almost an unnoticeable contact exposure with rabies contaminated material might be able to induce disease. And if you're in a cave with thousands of bats and a bunch of bat guano, my guess is that you got a chance of getting that type of exposure.
Yeah, and if you're scraping yourself over the rocks to climb through a cave, you may not notice another little scrape that happens to be a bat bite.
Right.
Back on this Wisconsin protocol, I thought that I didn't read the Wired article, but I skimmed it, and part of the hypothesis in that article was that maybe it was just coincidence that this the person who got the Wisconsin protocol might have survived anyway, and that might be the case for other survivors that are listed in that article and in the literature.
Right.
So, okay.
So now it is in Wikipedia. It's referred to as the Milwaukee Protocol. It's developed by Dr. Rodney Willoughby following treatment of Gianna Geese, and it involves chemically induced coma and administering antiviral drugs.
Right.
I guess the coma is the idea is that it prevents virus from getting into the CNS. Would that be the idea?
It slows down the replication.
It slows down. I think the idea is to slow down the replication in CNS and also to reduce the brain swelling. something along
She
those lines.
She was given a mixture of ketamine and midazolam.
That'll do it.
Right, so they put her in a coma, which is done medically for various reasons, but in this case, the idea was to try and give the immune system a fighting chance.
So 6 days later, she was brought out of the coma, and she was okay.
And
They dropped her body temperature also?
It's not.
I don't think
say
so.
Here. No, it
don't
doesn't say
think
here.
they did.
Well, then how does that slow the virus replication rate down?
It says that it protected her brain until the immune response could kick in. So somehow it's not getting in as readily. And I don't know why a coma would do that.
I don't
either. He had a
misinformation.
hypothesis in this article.
In the Wired article?
Yeah, I'm trying to find it now. But it had something to do with slowing replication. Doesn't sound quite right to me, but
entry into the brain would make sense.
Well, maybe in coma the core temperature drops.
Let's see. He had developed an understanding that rabies doesn't kill by destroying neurons or causing inflammation. Instead, this theory went, rabies primarily affects neurotransmission, the electrochemical communication in the CNS. Through a mechanism called excitotoxicity, the disease sends the brain into overdrive. And so he reasoned that the battle against rabies might be a battle for time. So if he put her into a coma, that would suppress the brain activity, and they would put her on a ventilator and whatever, and give the immune system the time that it needed.
Right,
but then
the idea is that it's the neurological dysfunction of the infection that's killing the patient, not actual cytopathic effect or death of neurons or anything.
Wow.
Right.
So put them out and get through that till the virus clears.
Right, keep the neurological problem from manifesting itself, hopefully, and let the virus clear.
So it looks like there's been, I saw somewhere that something over 50 patients have been now treated with this protocol, and there's been about 4 survivors. So.
Right. And the hypothesis.
10% is better than 100%.
Right, but the hypothesis in this article is maybe that they would have gotten better anyway.
Yeah.
Right.
Well, except that the history with this is that you present, the history, at least the dogma that I'm presented with, is that if you show up with symptoms from rabies, it has a virtually 100% mortality rate.
Yeah.
Right.
So if 10% people are surviving with this protocol, that's the suggestion is that it's actually doing something.
Yeah. 14% of dogs survive, bats survive, and if rabies doesn't kill 100% of its animal victims, maybe it doesn't kill all of the human victims.
Maybe not. Maybe 100% is an exaggeration.
That's what this Wired article is positing.
Okay.
Yeah.
Kathy, can you take the next one from Mickey?
From Mickey. Hi, guys. Interesting stored article on Fox News about vampire bat bites conferring resistance to rabies. What are your thoughts on the subject? And there's a link. And then, great show. Thanks for all your hard work. P.S. Have you thought about doing a show about rhabdovirus that would include a plant rhabdovirus scientist? And she mentions Andy Jackson or Michael Gooden. So, this Fox News article about vampire bats, I think I didn't check this one out. Sorry.
Okay. So this is based on an article which was published a few months ago in American Journal of Tropical Medicine and Hygiene. One of the authors is Charles Ruprecht, a rabies guy at CDC, who has in fact written the show before. Basically shows that in this Peruvian Amazon population, a number of the individuals have antibodies, neutralizing antibodies against rabies virus. 7 of 63 serum samples they got had this activity. Okay, and these individuals were picked for this survey because they had been reported to be bitten by bats. So in an interview, they said, yeah, I was bitten by a bat. So they took serum from them, and 7 were positive for neutralizing antibodies. So it's not that they're resistant to rabies. We don't know that. They could be, but they have neutralizing antibodies. And that's interesting because if they were bitten by a bat and infected, then why aren't they dead?
Right.
So But, of course, it could be a cross-reacting virus that we don't know about, that's not been isolated, that doesn't cause rabies. Who knows?
Right.
Could be a lot of things. It's an interesting study. So there was a bit of attention given this in the press. So maybe it's not 100% fatal, Rich.
Maybe not.
Who knows?
Maybe not.
Mickey, this letter was from Mickey.
That would explain a lot of things.
Yes.
This letter's from Mickey, who is in the Simon Lab at University of Maryland. So this is a plant virus lab. I'm glad that you're listening to TWiV, even though we don't do many plant viruses here on TWiV.
Yeah.
But it's a good idea. Next week, actually, I'm going to do a TWiV from Nebraska. I'm going to have a plant virologist on the panel.
Oh, good.
Oh, good.
That's going to be T. Jack Morris. So that'll be fun.
Cool.
Let's see, Dickson, do you want to do a short one next? Because this is from John, because the one after this is really long.
Okay. And it's still on the rabies subject anyway.
It sure is.
So John writes, rabies seems to be in the news these days. There's also a new cultural history of rabies entitled Rabid by Bill Wasick and Monica Murphy. I'm listening to it on Audible, and it's fascinating. Regards, John Wurzelman. Has anybody else encountered this sort of popularization of a virus that we're all keenly interested in?
I think they're all in the news these days.
All of them. Every one of them.
Well, this week there's this new rhabdovirus. There was a new coronavirus earlier this week.
See?
The press is
and the
fascinated.
West Nile has been in the news.
So this is a Times article which is about the Peruvian study, which we just mentioned, by Donald McNeil. Antibodies seen in Amazon dwellers suggest that rabies may be survivable. There we go. He put it in one headline, which we talked about for 5 minutes.
Yeah, the guy's a professional. He's a professional. Come on.
That pretty much sums it up.
Nice bat picture, too.
Very nice.
Love it.
All right. The next one is from Cristiano, and the subject of his email was, "Greetings from the end of the world, Dunedin, New Zealand. Dear TWiV hosts, I caved in when my 4-year-old daughter asked me for an iTouch last year. I resisted for as long as a father could, but in the end I gave up and bought her one. A few months later, she cracked the screen, lost interest in it, and asked me for an iPad instead. Initially, I laughed at her request, but since I caught her mother looking for an iPad ad in the weeks preceding Mother's Day, guess what? Yes, you're right. I caved in again and bought them both the latest generation iPad. I noticed there was nothing wrong with the iTouch except for the cracked screen, so I decided to fix it myself. A few attempts later, and the broken iPod was good as new. What am I going to do with this, I thought, realizing that the whole world was ahead of me with regards to popular electronic gadgetry. Play games, said my daughter. Listen to podcasts, suggested my creative wife. How do you do that, I asked. That question has thrown me to an entire new world. From then on, it was just a matter of hours until I found your program, and I truly fell in love with it. Oh, he says don't say that out loud on the program. Oh dear, sorry.
Too late.
I'm a virologist as well. I started studying baculoviruses when I was an undergrad back in Brazil and eventually got a BSc degree from the University of Brasilia. Soon after, I went to Japan to study more about baculos and pursue my postgrad degrees, MSc and PhD, at Nagoya University.
Wow.
In 2003, once I could safely display the Mission Accomplished banner, I sailed away to the uncharted waters of New Zealand for a short postdoc position at the University of Otago in Dunedin.
Dunedin.
How do you say it?
Dunedin.
Dunedin? I'm
Yes.
Dunedin, yeah.
I'm Dunedin, that's right.
Yeah, Dunedin.
Dunedin. Okay, thank you. A place I now call home.
Great. He must be multilingual. Look at all the places he's been.
Yeah, you can probably speak Japanese and Kiwi.
And Portuguese.
After my postdoc experience, I became interested in the field of science communication and documentary filmmaking. Was fortunate enough to have stumbled across a postgrad course at the University of Otago, which combined these two topics. For my final project, I decided to combine my virus research expertise and tell the story of the Auckland Island pigs. And it gives a link to the movie. Movie posted by co-director Adam Thompson talks about the pigs being commercially exploited as promising xenotransplantation tools for the treatment of type 1 diabetes. Long story short, a couple of 100 years of complete isolation made the pigs germ-free. At least that's what the charismatic mayor of the city, where the pigs are now kept, and the biotech company tells us all. But the truth is far from it. The pigs have very low copies of PERVs, porcine endogenous retroviruses, integrated in the pig's genetic makeup. Mm-hmm. I would like to invite you, dear TWiV hosts, to watch the short movie for some feedback and perhaps discuss PERVs in the future. In addition, I would love to know your thoughts regarding xenotransplantation and viruses in general. That would mean the world to me, as even after having finished this movie, I still can't reach a verdict. Do we abandon this life-saving technology due to its potential risks of pandemic proportions, or embrace it and utilize it in a large-scale xenotransplantation worldwide? Please keep up with the outstanding work you guys are doing with the program. Your ideas and comments are truly inspirational. P.S. Really sorry about the animations in the pig movie. We received no money for this project and had to be creative with explanatory animations. And PPS is another movie he made, he links to. It only touches the issue of varroa mites as carriers of honeybee viruses.
Wow.
So I want to do the movie review part.
Go ahead.
Because I thoroughly enjoyed it. It's got really cool scenery, plants, birds, and other animals, some really nice sub-Antarctic footage. The Mayor Shadbolt, the mayor of Invercargill, is just a stitch. You could watch it just for his monologue. And I found the animations charming.
Oh, wow.
So
I agree completely.
2
4
thumbs
stars.
up?
It's incredible. The mayor is a hoot. He really
He's
ought to
a
go
hoot.
on.
I've only watched the opening sequence of it before I had to check in for TWiV, so I have that ahead of me.
Yeah. Watch the whole thing. It's worth it.
I thought the animations were fine. If he hadn't told me it was low budget, I would have said, they were trying to be creative. You know, 'cause they use paper and writing on the paper.
Oh, sure.
That was
It's fine.
terrific.
Sure.
It's fine.
Sure. Nice.
Well, people do use pig parts for transplants already.
They do.
Yeah.
They use heart valves, right?
That's right.
Yep.
And there are PERVs in the genome, and so far they have not been an issue.
Right.
That's my understanding.
But now,
And I even found a study where they did a long-term study in immunosuppressed transplant recipients, and they didn't find any PERVs in them. I mean, the viruses, the PERVs, the pig endogenous retroviruses, these are non-defective. They will infect human cells, but even in these conditions, there don't seem to be any issues, which is not to say that there won't be in the future, but...
Yeah.
Go ahead.
Yeah.
I'm sorry, Rich. Go ahead.
when you transplant heart valves, is that actually living tissue, or is that more like cartilage or something like that?
Or connective tissue.
I don't know.
It's not. It doesn't have. My understanding is that it does not have cells in it.
That's correct.
Okay.
That is correct.
So it's more of a cartilage type of structure. And
Right.
the two choices for heart valve replacement are you either get an artificial valve or a pig valve, and you'd much rather probably have the pig valve because then you don't have to be on Coumadin for the rest of your life,
That's
which
right.
is
Right.
a pretty unpleasant drug, I understand.
So when we're talking about xenotransplantation of living cells that are producing insulin, that theoretically could put you into a different realm.
But that's already
So
been done.
now when people talk about xenotransplantation, they're talking about livers and kidneys and things like that. Obviously, this is a much more potentially infectious situation. I have covered this story intermittently, and I haven't developed a strong aversion to the idea of xenotransplantation, but the thing that keeps coming up is if we switched organ donation to opt-out instead of opt-in, we wouldn't be having this discussion.
True.
Well, this is particularly something, though, for diabetes. So they're talking about islet transplantation.
Right.
And the link that he gives, the PubMed link that he gives, if you then click on the top related citation in PubMed, the title of that is the International Xenotransplantation Association Consensus Statement on Conditions for Undertaking Clinical Trials of Porcine Islet Products in Type 1 Diabetes. Chapter 5, Strategies to Prevent Transmission of Porcine Endogenous Retroviruses. Needless to say, I did not read all of this, but the point is that they're looking at this. I mean, it's not like they're going in and not thinking about it. And they have some ideas about screening in advance for tissues that are low, have low copy numbers of these endogenous retroviruses, and maybe eventually getting to a point where you could, declare them not an issue. So.
Yeah, and I think it's great that people are having these discussions and approaching it that way. But it's worth remembering that the zoonotic infections that really are dangerous to us are things that come from people hunting or a wild animal bite or all the stuff that we can't control in the world. So this is something that seems like a much lesser threat. You've got a very well characterized domestic animal that you're going to do a xenotransplantation from, and it doesn't seem. It seems like the concern may be a little bit overblown, but I appreciate that people are following it.
Mm-hmm.
Right.
And my understanding is that there are too many copies typically to be able to get rid of them, by various targeting
I would think so.
techniques. Yeah.
Yeah, it's endogenous retroviruses. Everything has them, and fairly typical in pigs. I gather they're just all over the place.
Yeah, so the question is, are those a problem? There are experiments that you can do to at least try and address that, and it's good. Sounds like they're being done. That's good.
But they do make virus, and those viruses can infect human cells. The receptor is there, so
Yeah.
you have to be careful. There is a paper,
and there can be, and they can recombine.
Right.
So there's the
Right.
opportunity
This-
for novel viruses.
or
This
go ahead, Kathy.
So this article describes that the real problem is one called PERV-C, and the recombinant viruses between A and C could be eliminated by using pigs not containing PERV-C in their germ line. And selection of PERV-C-free animals, if possible, could reduce the risk. So they're thinking about ways to do that.
Right. And also, it's worth noting that I don't think anybody is talking about using food pigs as donors for this sort of thing. It would be completely separate type of facility where you would maintain these animals, and they would not be in contact with regular agricultural animals that are being transported around.
Yeah, in
Well,
fact,
and that they would probably start with these Auckland pigs.
Well,
Yes.
they elsewhere people are developing pigs with the MHC genes knocked out, so they will transplant more readily.
Right.
They won't be rejected, right?
Right.
So
Right.
that's-
The other problem with xenotransplantation is the rejection issue.
Sure. Yeah.
So if you knock out MHC, you may not have to immunosuppress. So I found a paper from 2010 in Xenotransplantation, which is a journal, and there they put porcine liver pieces into people while they were waiting for a liver transplant, and then eventually they got a liver transplant. But they monitored these people who were immunosuppressed for 5 years because they had a human liver transplant, and they found no evidence of PERV DNA or RNA in these individuals for 5 years.
Right.
But
And-
it's very interesting, the last paragraph. Is this enough to reassure us regarding clinical trials? And they say, we are chastened by the considerable angst generated by recent contradictory studies of XMRV.
Yes.
So there you go, Cristiano. I think that's a cool discussion.
Should I read the next one, Vince?
You just read one. Someone else's turn. The next one is Alan Dove.
I will take that one. Matt writes, "Hi, Vincent gang. Last week you had a letter from an Olympic athlete who wanted to know how he could stay in touch with science while training. I was amused when Rich said there is no way you could work for your PhD and train for the Olympics at the same time, because that is exactly what my student, Maria Mikta, just did. She was the only female race walker from the U.S. to compete in London. Now she's back in the lab as a full-time student. Training was balanced carefully with lab work over the last few years, but being right next to Central Park made it a lot easier. She even had a first author paper while training.
How about
Not
that?
bad.
Yeah.
Not bad at all. Pretty good.
Not at all. Yes.
Matt Evans, our Hep C friend at Mount Sinai.
Well, as we said, people who can do stuff like compete in the Olympics are disciplined. So
Yeah.
this is discipline squared.
Before we go on further, I just want to mention a word from the American Society for Microbiology, who partially sponsors TWiV. They are having a membership drive until the end of the year. And to find out about that, you can go to asm.org/advance. And I think most of us are probably ASM members. I know Dickson is not.
I am so.
You are?
I beg your pardon.
I don't think you are.
Yes, I am.
You're ASP, aren't you?
No, ASM.
As well. And Rich is probably an ASM member.
I am indeed.
Are you, Kathy?
Of course.
Probably not. Alan, though, right?
I am not currently.
Okay.
Aha.
I think I might have been at one point, and then it lapsed, and I didn't get back to it.
So I joined when I was a graduate student in the 70s, and I found that it's good to be part of the society. They run great meetings. They have great journals, of course, including the Journal of Virology. They have microbeworld.org now, of course.
And Infection and Immunity.
Yeah, great journals. And so they're a wonderful society for microbiologists. And if you're interested in microbiology, they have good student rates. They have now a really good student deal, which is quite cheap. So I would check it out. I think it's a great society. They do great things. Beyond the journals and the meetings, they have public policy issues. They comment. They have a communications department.
Yep.
Really cool society. So and a lot of members, over 35,000 members globally.
That's a lot. That is a lot.
So if you're interested in this stuff, check that out. And we thank them for their support of TWiV. Dickson, you're chafing to have an email.
I am.
Because take the next one.
I usually get left out of this because I don't have a monitor in front of me, so I just watch you read these things. But
hey, you know what the solution to that would be?
I know. Bring in my laptop. I know.
Take the one from KH.
Oh, I will. Okay. So KH writes, Dear TWiV, I'm doing a EPQ, Independent Research Qualification, taken at end of high school, on the eradication of polio. Vince, you know a lot about that, and whether the current attempt is worthwhile or not. I have a vague memory of it being mentioned in TWiV that it is advantageous for attenuated poliovirus to regain virulence, but haven't been able to find anything on it this anywhere. Could you give me some further information or direct me to where I can find it? Regards, KH. P.S. TWiV is fantastic. It was what inspired me to choose a virology-based topic for my EPQ and to apply for university courses, which include virology. Prior to listening, I had very little interest in it.
Cool.
Yeah.
Cool.
High school student.
High school student.
Yeah, that's great.
So, Vince, how would you answer that question?
So I believe what I said once on TWiV was so that the vaccine strains have mutations in their genomes that rapidly revert when they replicate in your intestines. So remember, you ingest the polio vaccines, you go to your intestines, they replicate, and within a day or two, the mutations revert. So there's a strong selection pressure in the gut to select for the small number of viruses with the original virulent mutation. So I had said that having the wild-type mutation is selected for in the gut, and it so happens that that mutation is associated with virulence in the central nervous system.
Right.
Because it happens in everyone's gut, as far as we can tell, it looks like this is good for the virus, at least in terms of replication in the gut. And not only does it revert, but all sorts of recombinants occur. You take a cocktail of 3 different serotypes, and they recombine among each other, and everything that comes out is now able to replicate really well. So there's a lot of pressure, and that's what I meant. Not virulence per se, but it so happens that the selection of this change is associated with virulence.
Which kind of makes sense, because if the virus could survive without causing the disease, without causing the paralytic disease, why wouldn't it? It's primarily an intestinal virus. Why does it have this?
Yeah, it doesn't
virulence?
have to get into the... I mean, the CNS is probably an accident, right?
It's a dead end.
Yeah, it's a dead end.
The virus isn't going to get anywhere from the CNS.
Yeah, the gut replication is what it's all about.
Right.
And so this mutation makes it replicate more efficiently in the gut. But it also apparently makes it replicate well in the CNS, and that's what gets you. So I got to put a couple of papers in the show notes that'll give you some more information on that.
Yeah, this one about the eradication program looks great.
Yeah, that's a really good one. And is it worthwhile? Yeah. You guys see this week Bill Gates made a big talk somewhere about, he says, by 2015, we're going to eradicate no more kids with polio by 2015, and eradication by 2018 will be declared.
This sounds similar to something I heard a while ago.
You know what he said in this article? It's an AP article. He said, eradicating polio is quite simple. You just have to get the vaccine to everyone.
It's simple but incredibly difficult.
Yes.
Simple things are not always easy.
Yeah, that's right.
No, they're not.
Well, he's really committed to it, which is good.
Yeah.
Good.
Because remember, the program started before Gates got into philanthropy, right?
Absolutely.
And it was really going to fizzle out. It was running out of money, and with him stepping in, this is really good.
The program started before Gates got into Windows. Seriously.
1988, right?
Yeah, this has been going on for a long time.
I think the really fascinating issue is post-eradication strategies. What do you do? With the vaccine strain circulating and reverting, and it's really something.
You can't just stop immunizing.
No.
That's going to be very interesting. But they're waiting to get to that point and then figure out what to do.
Well, it's going to take money, because money's the barrier to immunizing everybody against with the inactivated vaccine to start with. So
it's going to take money and a long attention span.
Yeah,
sure.
And the latter has been severely lacking in most national level eradication efforts as well. Which, I think it's worthwhile if we can get rid of this disease. That's absolutely worthwhile. But I wouldn't place long bets on that.
Okay, the next one's from Sydney. Kathy, we haven't heard from you in a while, right?
Okay. Sydney writes, "Good afternoon. I know you're busy, but I hope this email finds its way to you. I've been listening to your podcast for quite some time now. I really admire your innovation and ingenuity in regards to educating the general public about viruses. I've been interested in virology and host pathogen interactions for quite some time, especially those of newly emerging viruses. This led me to pursue my MS in entomology last fall. I have 1 year to go, and I'm beginning to look at PhD programs in virology in order to combine the two skill sets. Viruses like Ebola, Hendra, and Nipah are especially intriguing to me, and I'd really love to work with newly emerging pathogens. However, as I search for PhD programs, I'm finding that the number of researchers working in this area is low. It seems that the majority of this work goes on in level 4 laboratories, of course, by researchers rather than professors. I was curious to know if you could recommend any programs to me that might be a good fit, or if you could tell me whether working with another virus for my PhD would affect my ability to land a job with level 4 agents later in my career. Any advice would be great. Although I've almost completed my master's degree, I still feel like a nascent scientist with a lot to learn. You, Rich, and Alan helped tremendously, though. Have a great day, and I look forward to hearing from you. Sydney." Well, I think there are two approaches. There are likely some places where you could work on a virus at a pretty high level, maybe not level 4, but maybe a level 3 virus, and then move on to something in level 4 subsequently, or work on something that's level 2, get really good, solid training in virology as a graduate student, and then go to that level 4 type laboratory as a postdoc.
Yeah, I think if you wanted to do 4 work as a investigator, you'd have to do a postdoc in a 4, right?
Right.
So.
yeah.
And but as an entomologist going into virology, we should point out we know of at least one BSL-4 facility that has an insectary built in, right?
Yeah, that
it's
was
not
right.
quite open yet.
Once it opens, yes.
Right.
Yeah.
Right. But I mean, there are certainly places where graduate students could likely work on a virus that's BSL-3, or work in a BSL-3 facility.
Do you think it's useful? Do you think it'd be useful for somebody to specifically have BSL-4 experience as a graduate student, or is that something? I mean, to me, it seems like the science would be the key issue in the PhD.
Right.
From seeing how long things take in a BSL-4, I think you should do your PhD in a 2 or a 3.
Exactly.
That's actually a good point.
I mean, it just took, as you will see from the movie, everything goes slowly in a
Everything
BSL-4.
takes forever in a BSL-4, and one of the reasons there isn't a whole lot of published work on a lot of these viruses is that they are a huge pain to work with.
Kathy, wasn't there someone at Michigan who isn't still, who goes somewhere else to a BSL-4 to work?
That was Sonia Gerard.
Right.
Before the BSL-3 was available here, she was going back to CDC where she had done her postdoc with Stuart Nickel. But Yeah. So, but I can think of other BSL-3 labs, like Ralph Barrick's lab in North Carolina, West Nile virus labs that are doing BSL-3 work. So, again, I think getting good, solid training in virology where you can do it in an expedient way is a good thing, and then maybe move to your postdoc and work on higher level agents.
Although I think Elka did her PhD and postdoc in a 4 lab, right,
Yes.
guys? You remember that?
And I think at this point in this conversation, our first letter writer, Dennis, is probably jumping up and down.
Right.
And I think it's important to point out that the University of Texas Medical Branch in Galveston has a graduate program.
Right.
And a good medical
there are
entomology
probably
program.
opportunities out there to go to graduate school in an environment where there's access to BSL-4.
Oh, yeah.
Yes.
Right.
Yeah. So, I mean, this is our view, but Dennis may want to weigh in for sure. He may say, oh, it's fine to do a PhD in a 4.
Yeah.
Alan, can you take the one from Dallas?
Sure. Dallas, that's the person, not the city, writes, TWiVers, still going through old podcasts and don't understand viruses, but I do know a little physics on using the supercritical CO2 to create a dry virus. May allow viable virus particles to survive. The killing of envelope, which episode was this?
That's
Long ago.
long time ago. I think we talked about this to lyophilize, right, or something?
Yeah, the idea was to be able to stabilize a membrane virus so that it could be distributed as a vaccine without a cold chain.
Right.
So at room temperature. And this was a type of, quote, drying, unquote.
Right.
Yeah, so I think we said we didn't know how it worked.
Right.
And he's telling us.
Okay.
And this
So
is good.
he says the killing of enveloped viruses on drying may have to do with the surface tension of water changing the physical structure of the virus particle, and drying under supercritical conditions, the surface tension of the gas-liquid interface goes to 0. And then he provides a link to a Wikipedia article on aerogels, which are really cool materials in and of themselves.
This is true.
It links to that to see how an open gel type structure can be dried without destruction, just a common example that shows the principles. If the virus didn't change upon drying, it is small enough that the time it would take to rehydrate from the gas phase at 100% relative humidity at lung temperatures and lightly hydrophilic surfaces could be so fast that no structural collapse would occur on rehydration. Someone else probably already answered this physics type question. No, in fact, I don't.
We don't remember it if they did, so
Yes.
we're glad to hear it again.
Yeah. Cool.
Yeah, thank you, Dallas.
What's a scientific hatchery?
I don't know.
But
Dallas has written us before. I don't know if you remember, there was a email about shrimp farming. I think, or maybe that was someone else in Dallas.
There's something else. Did Dallas write? Is Dallas got another one?
Yeah, he
in
does.
this?
He does. Yeah.
Okay.
that may explain the hatchery.
I'm looking at his website, but it's not loading here.
Right.
He's got a PhD. He's in California.
Nice.
And if you want his cell number, I can send it to you. Rich.
Okay.
I think we're up to Nathan.
Yeah. Nathan writes, as was mentioned in TWiV 178, that Puerto Rico, it was mentioned in TWiV 178 that Puerto Rico doesn't currently have other mosquito-borne viruses like yellow fever or malaria. Can this be attributed to anything other than luck? Do any countries undertake efforts to curtail the spread of viral vectors by controlling used tire trade or otherwise? Thanks, Nate. Well, Dickson?
Well, yeah, everybody's pointing the finger to me now because you guys don't know and neither do I. But but my guess is that since we've had the introduction of Aedes albopictus from those tires from Japan back into the United States, that we're now much more careful in looking for hitchhikers on other retread products and things of this sort where standing bodies of water might exist. Puerto Rico, I guess, is out of the trade winds from Hispaniola, because otherwise they would have dengue at least, but they don't. So they are lucky. I think they are lucky.
Well, they have dengue.
They do
They
have
have dengue. Yeah.
Well, dengue is a mosquito-borne infection, and
But he's saying yellow fever or malaria. Why isn't there malaria in Puerto Rico, Dickson?
Well, because they have a good medical
Because
care
people would get
system,
treated.
and they get treated, and the mosquitoes don't harbor it as reservoirs from generation to generation, and therefore you need to acquire it every time.
Same with yellow fever then, right?
No, yellow fever is vertically transmitted in mosquitoes, so that's possible, and that's why dengue survives here too, I guess.
Okay. Yeah,
But yellow fever would have to be, once it's eliminated from an area, it would have to be imported.
Right.
And being an island, and Aedes aegypti, the vector, is certainly in Puerto Rico.
Yeah, sure.
And they, Kathy, you had linked to these 1963 reports with the elimination campaigns that were going on. I think that was part of the malaria eradication effort.
Correct.
That particular one was actually yellow fever, elimination of urban yellow fever in the Americas.
But there were a whole passel of related programs that mostly went about spraying DDT on everything to eradicate malaria. Don't laugh. This happened and obviously didn't work. Yeah, so that's the mosquito is there, but as far as vector control, I think every country that can afford it has vector control programs, and certainly every island I've ever been to has had some kind of inspection and has been pretty sensitive about what you bring in and what you take out.
Are they still spraying on Qantas before you get off the airplanes when you land in Australia? Because they did when I was there. They don't want anything to fly out of that airplane except tourists.
Well, TWiV 178 was with Tyler Sharp, if I remember.
Yes.
And it was what he talked about, his efforts to control outbreaks.
Yeah.
Right.
Dengue, right?
Yep.
Right. And on the CDC recommendation before you go to Puerto Rico site, they don't say anything about yellow fever. And in fact, in another link I saw specifically. So, yeah, yellow fever, malaria, not an issue, but dengue, yeah.
That's right.
Yeah.
Okay. Who's up next? Tell me. Rich, are you up?
Didn't I just do that one?
I can do this. It's another short
That
one.
was Dickson.
Yeah, I just did that one.
Okay.
Go, Dickson.
Nathan writes, this question is in response to episode number 182. You three discuss the benefits of immunizing in the skin rather than the muscle. This may be a naive question, but wouldn't injecting vaccine directly into the bloodstream be better than both skin and muscle? Is this an issue of medical difficulty and patient compliance? Who wants that one?
This is, by the way, this is from my virology class. This is left over, but I thought it was a good one.
Okay.
We did a paper where they used skin immunization of mice to immunize them against flu. Remember this one?
Yeah.
Right. So I'll tackle it a little bit. I think it's not really an issue of medical difficulty or patient compliance. It's an issue of probably what gives the best effect for immunization. And if you inject into the skin or the muscle as compared to the bloodstream, you will have a localized response, and you'll have the local antigen presenting cells picking up the antigens, taking it to the nearby lymph nodes, producing what you need for an innate response, which can then develop into an adaptive response. If you inject into the bloodstream, you don't have that sort of localized concentration issue. From the bloodstream, it's probably, I think, going to go to the spleen, and you would probably then get a response, but it's going to be a more diffuse one. So I think it's an issue of what works rather than what's easy or hard to get into patients.
Although, coincidentally, perhaps, it is also a whole heck of a lot easier to inject into muscle than into a vein.
Oh, yeah.
Yes, that too.
There's no vaccines are put in the blood, right, as far as I know?
No.
But because, as you
skin,
said...
I agree. Skin in particular is, I think, specialized to survey the environment for invaders, and it's crawling with cells that professionally go around and gobble these things up and present antigens to the appropriate lymphoid tissue. So skin turns out to be a very specialized for this function.
especially for these vector-delivered viruses, where a mosquito puts it in the very outer reaches of your skin. That's very different from an intramuscular injection, which is how you get the yellow fever vaccine, right?
That's right.
Boom.
Yeah, but the yellow fever is a live virus vaccine, though, right?
Well, live as viruses can be, Dickson.
I mean, they replicate, is what I'm saying. They call it a live virus.
I remember seeing a paper showing that there is a very different response for these kinds of vaccines when you put it, in the skin versus the muscle. For a vector-borne.
Well, that's the biggest problem they've had with the malaria vaccine. If to jump up to the eukaryotes just for a moment.
That's right. I remember that.
They had huge differences, and the natural way of getting it is by mosquitoes biting you, but then it gets into the bloodstream and goes to the liver, and then it gets into the blood. So there are these stages that it must go through first. It's very complicated, but I guess some viruses do that too, though.
So I have a rap about this that could go on for a little while regarding a paper that is just being submitted that I'm aware of. I can go on that rap now, or we can wait till it's published.
Is it from your lab?
No, from Dick Moyer's lab.
Well, you should probably wait till it's published, right?
We should probably wait till it's published.
Let's do that.
Yeah, we can. Once it's out, we can do that.
Because I can't wait till this gets published because I really want to talk about it. Okay.
All right, who's up now?
You are.
Yeah, how about you?
It's a really long one.
Good. Vince loves reading.
Bronwyn writes, Dearest TWiV hosts, Reservoir? I'll try to be as brief as possible.
I don't think so.
Though typing that rather than simply doing it is probably not helping there. Anyway, my name is Bronwyn. I'm a second-year biomed undergrad studying on Australia's Sunshine Coast, and though only newly infected, I'm immensely fond of your show. I love the breakdown discussion of papers and new research, which saves me drowning in the great sea of articles that is Scholar/PubMed/SciDirect, etc., etc., etc. As an undergrad wanting to get into research, number 184 on the broken science system, and more importantly, how to work towards fixing it and just survive in the meantime, was brilliant. I clicked download on the Casa de Val and Fang papers as soon as I had played it out, which meant that they had just finished by the time I finished playing it through the second time over. I guess he's got slow downloads down there.
Yeah.
Right.
I'd been getting despondent over the dog-eat, poison, cripple, render otherwise harmless world that science research seemed to have manifested itself as, a model one would have expected from a somewhat business-like paradigm, not the province of a collaborative pursuit of understanding, so that we, as a biologically social species, might go about interacting with the world in a more intelligent way. But enough ranting. Essentially, I love your work, guys, and incidentally, it is about 20 degrees centigrade and a bit overcast here and there, which is really quite cold for us. Winter just started. But now to get to the reason why I'm spamming your already inundated inbox. Earlier this term in a micro class, my lecturer was talking about plant viruses, and I couldn't help noticing they, for the most part, have RNA genomes rather than DNA. I was curious and asked, but she was unaware of any reason why and couldn't find any explanation when she researched it a bit later. Either could I. Any ideas? Is this possibly a quirk of nature arising from an RNA-containing evolutionary forebears, or does anyone know of there being some sort of selection preference for RNA genomes in plant viruses? All right, I'm going to read an answer to that, and someone else can resume reading.
Okay, I'll resume reading after you read your
So
answer.
I sent this to T. Jack Morris, who's a plant virologist out in Nebraska with a lot of years of work under his belt. And he responded, I can think of no fundamental reason for this to be the case other than an accident of evolution. Here's the rationale. It turns out there are no true double-stranded DNA viruses in plants, I guess he means. There are pararetroviruses like cauliflower mosaic and single-stranded DNA viruses like Gemini viruses. So it is not because plant cells could not support the true DNA virus replication. It's not likely a cell-to-cell movement restriction because there are plenty of viruses of papilloma size that infect plants. It could have something to do with the nature of plant meristems, where cell division is primarily restricted. It is a question worth considering, perhaps at the TWiV. And he means the TWiV over in Nebraska. So maybe we'll continue this in Nebraska, but he doesn't seem to have a immediate answer.
So does the Midwest become the TWiV over then?
Cool. Go ahead, Dallin.
All right, I'll continue the letter. Also, I was watching a video, MIT World talk on global pandemics, November 17th, 2009, given by Hida Plug, professor of biology at MIT, available from iTunes University. It's a soapbox talk night sort of thingy. Anyway, Hida briefly discusses the FDA-approved method of flu vaccine production, pointing out the fact that not all flu strains grow equally well in eggs, which limits the strains covered by an American-made vaccine to those that perform sufficiently in chicken eggs. Is this a serious limiting factor in the vaccine quality, or merely something that needs to be kept in mind when making a vaccine? In Europe, flu strains are grown in cell cultures. This would eradicate the problem of allergic reaction caused by chicken proteins. Is there any other reason why an allergic reaction would occur in response to a vaccine?
You want to deal with that first?
Yeah, there's now a cell-based vaccine that is approved in the U.S., right?
About to be, I believe.
About to be, yeah.
But what he says here is not correct. I don't know if Hida said that,
right,
or that's Bronwyn's interpretation, right? It's we make reassortants. So many clinical isolates don't grow well in eggs, but we make a reassortant with a good egg-growing strain,
right,
and transfer the HA and NA genes into that. So that there's no limitation of what strains can be included in a vaccine.
Well, I mean, you could say that the strain that's included in the vaccine is not exactly the same strain that's isolated clinically, but it's immunologically the same strain, and that's what matters for the purpose of the vaccine.
Right.
So that's not a serious limiting factor. The egg allergy thing is a relatively minor issue. It's apparently a pretty rare allergy, but it is a potential issue in the vaccine, and it's a major reason, one of the major reasons for going to a cell-based system. Okay, continuing. If you have time, I recommend watching the talk. There's a great dismissal of an evil drug company flu vaccine hiding conspiracy theory, and a criticism of the economic priorities of funds allocations in research, and of the concern paranoia the U.S. seems to have over biodefense, despite the lack of reasonable threat assessments. Just think.
Paranoia?
Paranoia.
Will destroy it.
Just think, this was 3 years before the NSABB censored the Kawaoka and Fouchier research. But my favorite part, if it's not the fact that he's wearing bright red pants, is the final word he gives on the moral obligation of vaccination and the dangerous selfishness of people who rely on herd immunity. It's a serious problem, I think. Here in Oz, Australia, there's a massive anti-vaccination sentiment rooted in a distrust of science, medicine, drug companies, and a fear of vaccination side effects like autism from a measles vaccine. Geez, looks like a symptom of the winner-takes-all system with its pressure to publish giving rise to bad science and fraud. A retraction does not and cannot undo the damage done by bad science. This attitude seems to be concentrated in more alternative towns like Byron. Paul Corbin, director of New South Wales North Coast Public Health, said there had been twice the level of disease in Byron Bay as had occurred in Ballina, a town pretty well adjacent to it. This info is taken from the ABC TV show Catalyst, Episode 9 of 2012. Is regional social psychology also a big factor in American vaccination rates? Do public health systems attempt to address this? I mean, wouldn't unvaccinated people, when concentrated into small geographic areas, be so much more dangerous than if they were dispersed, as the statistics for the unvaccinated to vaccinated ratio make it appear? I don't expect this to be read on air or anything.
Too late.
Now you tell us. I'd really just appreciate any answers, info, links, TWiV episode suggestions. I'm still working my way through the back catalog, just finished 2008, in addition to listening to current episodes as they're released, so I do not doubt that some of this would be addressed therein. I'm especially curious in regards to the apparent plant preference for RNA. Oh dear, short message fail. That was so much.
Okay, we like reading.
Of things.
Uh-oh.
You're breaking up. Alan, you're breaking up.
Am I breaking up?
Yes.
Yeah, we didn't hear that last sentence.
Okay.
Read it again, please.
Oh dear, short message fail. That was so much longer than the paragraph I had originally intended. My apologies and many magnitudes of thanks from down under.
Right.
So, Alan, can you address the social psychology question?
Yeah, to the extent that it's possible.
Isn't that what happened in California? You get these clusters of people not vaccinated against measles.
I'm just looking up now. Ashland, Oregon.
That's right.
Is a cluster.
All right, so the psychology aspect of it, that is definitely a big factor in the U.S. as well. That's not unique to Australia. And I forget who said this, but somebody commented that if you wanted to find the major areas where vaccination refusal is highest, you should just get a map of Whole Foods locations.
And Birkenstock shoe stores.
Yeah, there are probably other things that would correlate here as well, and it's it's a serious problem among certain social groups, and it is perhaps ironic that the social groups that do this are the ones that tend to be more affluent as well. So there's a particular demographic that is more likely to believe this anti-science about vaccines, and that obviously people cluster demographically and geographically. So yeah, you're going to have neighborhoods of unvaccinated kids, especially going to the same schools. And in fact, many of the school outbreaks that we've seen have been at places that have had a high rate of vaccine refusal and that have drawn from communities that match this type of profile. So yeah, that's exactly what's going on.
Yeah.
And yes, it's ridiculous for people to take this selfish attitude that, rely on herd immunity.
Right.
That's
Doesn't work.
That's absurd, especially when it's not as if you're really risking anything.
Here's an interesting article from the Oregon Health Authority that gives a map showing the immunization rates in different counties in Oregon.
Oh, wow.
So this has been a problem in some areas in Oregon. I'm just waiting for an outbreak in some of these places. They're doing an experiment is what they're doing.
Yeah, they are.
I'll paste this into the notes.
And in fact, Trina Sideris put together a map like this for Illinois, I think. Before I think she's no longer at the Chicago Tribune, unfortunately.
Right. Yes, she did retire. Yes,
their loss. But she did this very deep dive in the data and found school vaccination refusal rates and plotted it by school district. And it was really, really interesting because you found vaccination rates versus school district. You found lower vaccination rates in some of the poor inner city schools, and that was entirely economically driven. And then you found low vaccination rates, particularly low vaccination rates in some of the most affluent suburban districts.
Right.
So it's exactly this type of thing.
Yeah. I wanted to add something to the vaccine production scene here. I just recently got back from a meeting where I was amazed to learn that in Texas, at Texas A&M, there is a large multi-story building using tobacco plants to grow influenza virus vaccine, virus-like particles. And the blurb that you can check out from the Texas Plant Expression Vaccine Consortium website says that eventually they want to boost this up to 1 billion doses of influenza virus-like particles. That's major, all right? And that really gets around this allergy situation for duck eggs or chicken eggs or any other kind of eggs for that matter, or tissue culture. You get a higher yield, it's cleaner to begin with, and it just points up the fact that if you look for a solution long enough, you'll find it.
Right, and it's as healthy as tobacco.
Hey, there you go.
Dickson, if you search Google for vertical vaccine farm, you get TWiV number 47.
That's right. Exactly right. I actually went back and listened to it once I was told about this. By the way, that facility is a DARPA-sponsored program, and it's not available to the current public yet, but it will be because I also got an email from my friend Peter Hotez, who has been on our TWiP show several times, and he says, because he's now in charge of the vaccination center or the vaccine center that used to be in operation in, I guess, in Maryland somewhere, and he's very intimately involved in this. He says, we're doing that, and we're doing it better than that. So I'm waiting to hear back from him on that so I can give you a bigger and better report on this. I'm very excited.
Cool. All right,
Vincent, you got to read your quote here. I really.
Oh, yes. So
love
this letter reminded me of a quote from Blaise Pascal, who lived in the 1600s. He said, "I have only made this letter longer because I have not had the time to make it shorter." So it does take time to write short things. I know.
Yeah, that's right.
Sure.
But don't worry, Bronwyn, we appreciate every word.
No, it's the tapestry of a thinking person hard at work.
Rich, are you up?
Yeah. Well, I don't know if I'm up, but am I up?
Sure. Then we'll take Kathy next. How's that?
Okay. Dallas writes, just a quick comment on the podcast covering journal retractions and other scientific errors. A comment was made that the problems seem to be in the first-rate journals which have the most retractions, at least the ones we hear about. However, if you want to find a lot of papers that should be retracted, the second and third-tier journal have even more junk, advocacy, science, and false information. But most of it isn't worth anyone's time to bother proving false or pointing out the errors. To complicate the problem in the second and third tier journals, you have advocacy groups of various types, ranging from environmental groups to anti-global warming groups, playing a bootstrapping game to create scientific, quote, facts, unquote, with great truthiness that can be cited in the first tier journals. For an example of this bootstrapping, a friend of mine in academia was charged with the task of tracing the source of a scientific, quote, fact, unquote, about damage from the aquaculture industry on the environment that found its way in proposed regulations and into lawsuits. It turns out that an activist group created a damage estimate out of a consensus feel among other activists, and got it past reviewers in a third-rate journal. After adding a lot of caveats about this estimate just being a swag, that is, scientific wild-ass guess, with no data. The activist group then got another publication in a similar area in a second-rate journal referencing the first article without the caveats, which was then referenced in a first-rate journal by a related activist group with a common funding source. Being in a first-rate journal, it was now a proven scientific fact and used by regulators being lobbied by the activist groups. The industry was then facing a scientific fact with great truthiness, which didn't match their unpublished observations. With the high frequency of manure in second and third-rate journals, it may be a good training exercise for starting graduate students to plow through some of these journals and pick a paper to reproduce the most suspicious parts or demonstrate error in their methods or analysis. They would end up learning a lot about everything from good lab work to ways of analyzing the data. It may be a good way to sharpen the thinking of grad students and reinforce the message that all you read is not true. It would also force the students to go through a lot of articles looking for a particularly bad one for their class project and read them all very critically. A year later, when they're writing a paper, have to visualize how their classmates would cut it apart, and you may get better first drafts. If you want, I could provide several examples from the environmental literature, especially aquaculture, where truthiness rather than facts dominate the literature. I'm a semi-retired retread physics thermodynamics engineering type who spent 3 decades solving environments and aquaculture problem, heavy into using microbiology to destroy hazardous materials or convert waste products. It is all about the bugs and the bug habitat. Who is slowly working my way through old TWiV podcasts with great delight. Keep up the excellent work educating senior citizens about the fascinating world of virology. I have a great interest and little knowledge about phages in the real world, where they seem to often be the keystone species, not the lions and sharks discussed in ecology classes. Thank you, Dallas. P.S. In case you don't know, various shrimp do $1 billion+ in damage. Shrimp viruses, do $1 billion+ damage to the worldwide farmed shrimp industry every year. 90% of the shrimp we eat in the U.S. are farmed. Don Leitner at University of Arizona is the top man in the world on shrimp virus and may be an excellent guest. Good idea. Yes. The shrimp industry is far more sophisticated about identification, control, biosecurity, and managing virus than any other industry I know about. PCR testing at the farm level. I have been in shrimp hatcheries in the middle of remote areas of Central America that make our top-rated local U.S. hospitals look like cesspools of bacteria and viral contamination in comparison. Very tight biosecurity.
Here's scientific hatcheries.
That would be very interesting.
Our top-rated hospitals are cesspools of bacterial and viral contamination.
So we ought to do, we really do need to do these shrimp viruses because that would be interesting. It'd be nice to have an expert to do that.
Yeah.
I do have a comment about this, and that is that this idea of junk getting fixed in the literature by a chain of referencing is a real problem.
Yes.
And this is something that I think about a lot when I'm reviewing papers. Because when I see something come along that I think, even in a paper where mostly the data are probably okay, but a conclusion is drawn that's really not justified by the data, I think it's really important to get that taken out of the paper.
Yes,
Otherwise,
I
that winds up getting fixed in the literature as a fact, and now you got a problem.
There's a huge, long history of this where we end up with scientific facts that are wrong.
Right.
Correct.
Yeah. And by the way, most of it is not deliberate. So what he gave the example of is somebody actually using that phenomenon with an agenda. But most of this type of error just sort of quietly creeps into the literature, and somebody is a little lazy. They don't go back to the primary sources all the way, and they say, "Oh yeah, I saw that referenced in a paper," and then everybody references it. That's the truth.
So just an hour before we started recording, Arturo Casadeval, one of the authors of the Reforming Science papers that we talked about before, sent me a paper that is going to be published Monday in PNAS. So it's still under embargo. We won't talk about it. But it has all to do with retraction in the scientific literature.
Cool.
So maybe we'll tackle this on a future episode. Very interesting. He said the Times is going to have a big article on Tuesday. They're making a big deal of it.
I hope he's very careful with his data in the paper.
We had a famous immunologist here. I'm sure a lot of you have heard of him, Elvin Cabot. And one of his favorite journals that didn't exist, of course, was called Acta Retracta.
Yes.
Well, now there's a blog called Retraction Watch.
Yeah.
Really?
Yeah, that keeps track of all the retractions.
Very interesting.
Okay, Kathy.
Neva writes, "Howdy, TWiVs. This article in Ars Technica on disrupting the publishing model of papers in biological science might interest you and your fans." And it's a link. Keep up your valuable and entertaining podcasts. And Neva is the Neva in Buda.
Yeah. Glad to hear from Neva. Hadn't heard from her for a while. I was afraid she'd gone away.
Right. So I took a look at this, and maybe Alan did too. It's sort of an alternative open access publishing kind of model where each author would pay $99 up to 12 authors, but you could have more authors on a paper. I don't know. It's
one paper a year you can publish.
Yeah, you
right?
pay a membership fee, and then you can publish under that membership fee as many papers as you want that year in that journal.
I see.
Right.
Okay.
Right. But as a couple of people in the comments pointed out, whether this is really cost effective relative to publishing in a PLOS journal, for example, may be open to question. There were some other things.
So if you have 10 authors from your lab, you got to pay $1,000. That's it.
I think that's the gist of it. Yeah.
And they've taken a lot of shortcuts. They unload their archiving duties onto PubMed Central, So they just dump that on the NIH. And yeah, you pay for that one way or another, but they don't have to. I don't know. It's a lot of people are jumping onto open access publishing now. I think a lot of these things are going to fold. I think a lot of them have some pretty wacky business approaches, but some of them are probably going to stick around. And I don't know if this thing has legs.
One comment I have about at least recently encountered more than once with PLOS pathogens, these journals aren't particularly well copy edited, and some people aren't as compulsive about their writing and copy editing as I am. I get that, but it's frustrating for me to read a paper where there are so many mistakes that haven't been corrected by copy editing that I lose the meaning and the train of thought, and sometimes things seem to be the opposite of what they're saying. I've always thought that the ASM journals are so well reviewed and they have good copy editing to go along with it. That if we have more of these open access journals and people just basically write something up and put it online, it could make a mess of things.
Yeah. And I gather in this model, you pay your $99, you get to publish a paper that year. Nobody's commenting on the merits of that paper.
Right. They say that it's basically a PLOS One model.
Yeah,
right?
and PLOS One, there's some good stuff published in PLOS One, and there's some not so good stuff published in PLOS One, and so
I think that's
it's
okay.
A potential problem.
I think that's okay because I'm not sure that the journals should decide what's good and not. You should just let the reviewers do it. Right? And that's one of the problems with the top tier journals. You have a couple of reviewers deciding whether something gets published or not. But maybe the model should be if it's experimentally sound, you put it out there. It doesn't have to be groundbreaking, right?
Right. Well, and PLOS One is peer reviewed, right?
Yes, it is.
So.
Yeah.
But one of the criteria is not that it has to be a substantial advance to the field and such things, right?
Right. So it's a weaker version of peer review, and in a model like this, you do away with the position that would be called research editor, who's the person who normally receives the manuscript someplace like Nature or Science or Cell, and goes through them all, triages them, and figures out what's actually worth sending out for peer review.
Right.
So you do away with that whole gatekeeper function. I don't know. It's beneficial in the sense that the science gets out there quickly, but it's a potentially serious problem in the sense of the previous letter we were talking about. Here now you have a journal that can be simultaneously first tier and ninth tier.
Yeah.
So you can have a great paper published in one of these, and you can have absolute crud published right next to it. And what's the credibility of that journal? Well, the impact factor is going to be based on the best papers. So it's going to look like a very credible journal if you look at any of the objective measures of it. And yet there's going to be some real dross coming out of it that people can cite and say, hey, it was published in yada yada journal. And that makes it sound like it has a credibility that it doesn't in fact earn.
Good point. So the impact is based on citations?
Sure.
Not page views, right?
Right.
Okay. It's not an easy solution, but as they say in this article, disruption is going to happen.
Yes.
Right. Interesting. Well, we'll see. We'll see if it's got legs. Thanks for that, Neva. Dickson, you want to do a short one from Josh?
Sure. Josh writes, Dear TWiV doctors, you will probably get 1,000 emails about this, but there was an article in Nature about curing Ebola virus in monkeys. Here's the link.
That's it.
That's it. That was quick.
Since you don't have a laptop, you can't read the name of the article, Dickson.
You're right on. Well, it's nature.com/news/antibody-cocktail-cures-monkeys-of-Ebola. Ebola 1, actually. It was Ebola 1. But there's a quote below this in the news and comments by Dennis Burton, an immunologist at the Scripps Research Institute in La Jolla, California, says that the results are very encouraging, in quotes, for cases where the antibodies can be given within 1 day or so of exposure to Ebola, but the work is unlikely to help in cases where the infection is established and symptoms have appeared. Quotes, I would remain rather pessimistic given the explosive course of Ebola virus disease, he says. Everybody agree with that? Or
Yeah, Kathy was the one who pasted in that quote from the news and comment.
Right.
Mm-hmm.
Right. Okay.
So.
Yeah, it's treating monkeys with antibodies.
Yeah, so it's basically passive immunization, a classical treatment with, I guess, some antibodies that have been produced. And it highlights the thing that we've talked about so many times before here, and that is that a lot of antiviral treatments, by the time you come in with symptoms, is too late.
Right.
Mm-hmm.
But they are working on an Ebola vaccine, so presumably, if they ever use this, it would be before we had a vaccine, which is now, right, Dickson?
Yes.
And who would you give the vaccine to?
People working in P4 facilities.
Yes, of course.
That's a good problem, right?
Or
anthropologists. If you have an outbreak...
This is not a common condition.
No.
If you had an outbreak, you could contain the outbreak.
Yes, that's right.
Yes.
If you had an outbreak in Africa somewhere or somewhere else, you just immunize the community, but you wouldn't give it to people in the U.S.
And the fact is that this passive immunization, if you're if I were working with the Epidemiologic Intelligence Service and I flew into Africa somewhere where there was an Ebola outbreak and I got a needle stick, I might want to have this stuff around.
Yeah.
Sure.
Okay. Well, let's say you're working in a BSL-4 and you've infected some primates with Ebola and you're doing a necropsy and you cut yourself right through the glove into your hand while you're dissecting the animal. You'd like to have these antibodies, wouldn't you?
You betcha.
I'll take that.
Yeah.
You betcha.
Well, I seem to recall that for rabies exposure, it used to be that you would get a gamma globulin.
Yeah.
That's still part of the treatment.
That's still done.
Wash the wound, inject gamma globulin right into the wound, and then start the active vaccination.
So I brought it back to World Rabies Day.
There you go.
Good job.
So, Kathy, can you read the next one since you got a short one there? And Sarah.
This is the Sarah writes.
Yeah.
Hello, TWiVologists. The June issues of ASM's Microbe magazine had a feature article on virus-induced obesity that had so many holes I had to break out my pen and write all over it. I followed the references to the 2005 paper that the article seemed to be based on, and as much as I believe the possibility of an infectious factor in obesity, my BS meter just kept going off here. Looking past the incredible number of variables that weren't even considered in data collection, questionable sample handling, missing context, and leaps in logic, I couldn't get past two fundamental issues. One is the author's apparent belief in a causal connection between the initial detection of this virus in 1978 and an epidemic of obesity that seemed to begin around the same time. Detection and emergence of a virus are two quite different things. The other is that the glaring conflict of interest evident in his founding of a company that makes assays to detect this virus for consumers.
Right.
The company's website skips scientific caution and flat out states that AD36 causes obesity. The author is also the editor of the journal in which the 2005 paper was published. How awkward is that for peer reviewers? I'm coming off of 3 recent influences on the importance of keeping a skeptical mindset and using critical thinking as a verb. The dogged insistence of Mike Osterholm's statements during the NSABB brouhaha, reading The Panic Virus, and then reading A Demon-Haunted World by Carl Sagan. It seems like freight trains of misinformed, truthy opinion can leave the station before anyone figures out what's happening. And by then, all any rational person can do is stick to their evidence-based guns and let the situation run its course. Parentheses. I'm not stating that Osterholm was being truthy like Jenny McCarthy, but I've wondered whether he thinks that someone in the biowarfare labs in Iraq are just waiting to get their hands on Boucher's paper. End parentheses. My husband teases me because I don't mind writing a letter about something on which I have an informed opinion, whether it's about a product, customer service, a news article, etc. He has a point. Sometimes it's better just to let it go. There are limitations to all experiments. But when you all come across papers like this that make you just cringe, what do you do? I guess that was a you all. When you all come across papers like that.
What do you do? What do you do?
Is it better to let it slide, write a critique, or just talk about it on TWiV? As I move into a healthcare career, I'm concerned about these issues not only for the sake of science, but for the sake of public health policy, grant funding, and what new damage could be done with the combination of the media and outspoken narcissists or misanthropes. I now not only see microbes everywhere I go, but also the subtext of human behavior. Gray hairs may be soon to follow. Just kidding. Keep having fun. So this story about adenovirus 36 being related to obesity goes back quite a long time, and I do agree that the way the article is written seems to make a connection between the detection of the virus in 1978 and an epidemic of obesity. In particular, the fact that there's an increase in obesity in the developing world, which wouldn't necessarily be explained by junk food and other things. And so the hypothesis is that, that must be then perhaps because of an infectious agent. The detection and emergence are two quite different things. I agree with that. And then she does point out several other things about this article in Microbe, and I went and looked at the article myself, and I hadn't really realized this, but these articles in Microbe, which is the ASM sort of general journal, they're not referenced in the same sort of way that a primary research article is. In other words, there's a list of references at the back, but all of the statements throughout the article aren't specifically referenced. And I found that particularly frustrating. And so, where she said she found holes and had to break out her pen, I was just wanting to say, well, where did they publish that? What are the data for that? And so that was more of my reaction to it. There is a reasonable body of work that's published in this area about the connection between adenoviruses and obesity. It's very hard to demonstrate causality, as we know. As we start to know more about the interaction between viruses and the microbiome, that may inform what we learn about this in the future. So that's what I have to say about it from the adenovirus standpoint. Some of those other things maybe the rest of you want to address that she said.
Well, she's got this point here. When you read stuff like this and you cringe, what do? Well, we TWiV.
Yeah, I think writing to TWiV is a good idea.
And I blog about stuff like this.
Yeah.
Right, I do.
Because I sometimes I come across some little kerfuffle that makes the news, and I see lots and lots of crummy papers come across my email inbox and my radar in various ways, because I get press releases about everything. And most of it, fortunately, disappears into well-deserved obscurity. But every now and then, something like this gets picked up and turned into a thing. And if it bothers me enough, I'll put up a post and say, "Hey, everybody, here's what's wrong with this."
It sounds like Sarah is a scientist, doesn't it?
Yes.
Sarah, you should blog. If you could write to us and we'll discuss it, but you might feel better blogging and having other people read it as well. Although we have a pretty good audience here, I think all scientists need to do their part in this. And if you write a blog post now and then, that helps.
So we did a TWiV on this.
We did.
Yeah.
TWiV 101, Sizing Up Adenovirus.
Yes, I wonder who came up with that title.
And so it'd be interesting to go back and I forget, at the article, whatever article we did is there. It'd be interesting to go back and see what we thought of that.
Has there been anything more recent, Kathy, in this that's more compelling?
I looked at that earlier today, and now I can't recall. Sorry.
If you do in the next few weeks, maybe we could revisit it if it seems worthy.
The sizing up adenovirus was the adenovirus crystal structure.
No, there's also an article, Adenovirus 36.
You're right, the role of
obesity in children.
obesity. Yep, you're right.
This is a paper that does not have this individual on it.
Right.
Gabbert, Donahue, Arnold, and Schwimmer in pediatrics. I'd have to look at it again and listen to the episode again to refresh my memory on that.
I think we're pretty critical.
Yeah.
But Sarah, maybe you want to go listen to that. Although it might get you more burned up,
Yeah, there's at this point there is a lot wrong with this theory. There's a lot that doesn't fit. So.
All right, let's see. A couple of quick ones, and then one more. Daniel sent a link to a CNN article entitled "Taliban to U.S. End Drone Strikes in Pakistan or No More Polio Vaccines." Actually, you have blogged about this a bit, right, Alan?
Yeah, a little bit. So there's this whole thing going on. The raid that killed bin Laden, in the run-up to that, one of the things that they did to try and determine that was in fact bin Laden's house was try to get blood samples from some of the kids that lived there. And if they could prove that if they could get DNA samples and prove that these were bin Laden children, because they have reference samples from the bin Laden family, then that would be compelling evidence to go ahead and do the raid. And one of the mechanisms that they used to do this was the CIA organized a hepatitis B vaccination campaign, which set up in the town and did a couple of things and then tried to hopefully take samples from the kids in the house. It ended up not working, but even though they were still a little unsure about it, they went ahead and did the raid anyway, and obviously we know how that turned out. And since then, that CIA fake vaccination campaign, as it's been called, has been cited as a cause for some subsequent conflicts. And this article on CNN is about one of the really tragically stupid things that the Taliban said, apparently, that if you don't stop drone strikes in Pakistan on suspected Taliban locations, will stop letting the WHO officials come in to give polio vaccines, which is just, this is to benefit your kids, and why are you?
This is like taking a bunch of women and children and chaining them around a target,
precisely
a military target. It just makes you sick.
Yeah, and I did a blog post about my own kind of conflicted feelings about this whole thing, because the CIA did in fact do this dishonest Hep B vaccination campaign, and it's problematic, but polio vaccination is not the only priority in the world, and ultimately I'm not quite willing to condemn that effort. Is where I come down on it.
Right. All right. The last one today is from Judy, who is our science teacher fan in California. She sends a list of top 10 free apps for scientists from the Berkeley Science Review. They include Twitter, ACS Mobile, American Chemical Society, PLOS, RSC Mobile, another abstract app, MRS Bulletin, YouTube, Nature, Springer, Science Direct, and Science Facts. Most of these are publishing app except for that last one, Science.
Yeah, that last one looks like it might be fun.
So thanks for that, Judy. And unfortunately, we have lots of emails left, but I think we'll have to save them for another time.
I have a brief follow-up on the what's new in adenovirus and obesity. I use different search terms from what I used this morning. The first two papers are publications in PLOS One, as is the fifth one. So those are all 2012 papers. The first one is a meta-analysis of studies, and the gist of it is the relationship between AD36 and obesity should be assessed by further studies, including well-designed prospective studies, to gain a better understanding. So, yeah.
Jury's out.
Yes.
You bet.
Wow, we have a lot more emails here.
I know.
Have to read faster next time.
Oh my gosh. It's just like we didn't even do half of them.
Correct.
We did about half of them.
We did?
Yeah.
Maybe the ones left are longer. That's the reason. All right, let's do some science picks. Alan, what do you have for us?
I have the complete papers of Alfred Russel Wallace, which are now online.
Nice.
This is a project that, let's see, National University of Singapore is involved with it, and I think it's one of these where somebody who really, really just decided this ought to be done got it done. This information has been scattered all over the place historically. But now you can go and you can look through all of Wallace's publications and look at a catalog of his specimens, stuff that he collected, and of course read about him. And this was the fellow. He was a professional collector based in Southeast Asia, who sold curiosities to the Victorian market in the 1800s, and he came to pretty much the same conclusions that Charles Darwin was reaching around the same time. So as Darwin was working away and doing his experiments, Wallace was working away and doing his collecting for profit and coming to similar conclusions. And then it's sort of a Newton and Leibniz story, except that the Newton character, who would be Darwin, was a much nicer guy. And actually went out of his way to try and make sure that Wallace got some credit for having developed a similar theory.
Wow. Illustrations, too. Nice.
Yeah.
Great. Thank you, Alan. Rich.
Well, I've already leaked my pick. It's World Rabies Day. And I've got a link to it. There's a site here that's basically information on rabies and et cetera, and you can have a look through it. And I think we've done it. We've celebrated World Rabies Day in style.
Yeah, we had a bunch of rabies males.
The video that's there is really worth watching. Okay. It's a small child coming in to talk to a veterinarian, but they cover things really well.
Great.
It's worthwhile.
And it's also available in French and Spanish.
Oh.
Very nice.
You can buy rabies merchandise. Shirts, posters, logos, stickers.
Rabiesabilia.
You bet. All right. Well, our next day will be polio in October. Someone will have to pick that. Kathy.
So I chose something that was sent to me twice in the last week, and it's yo-yos in space. And it's one of the guys in his downtime in the space station shows his yo-yo skills and talks about the physics of it, and then talks about how the women who might be able to talk about the physics of yo-yos could impress certain guys, and the ones that don't seem so interested, well, then you're not interested in them anyway. But all the while he's doing these really funny, interesting yo-yo tricks in zero gravity. So
excellent.
He is clearly a yo-yo aficionado.
Yeah.
I love that he spins the yo-yo up and he holds it so you can hear the noise of it. And he says, "Those are precision ball bearings you're hearing there."
Terrific.
And he gets to name his own tricks that he creates in space.
Of course.
Cool.
I love it.
Thank you. That's great. Dickson, do you have a pick?
I do. It's called The Art in Science, and it's a website. And it's got some fabulous scanning electron micrograph pictures and lots of virus pictures as well. They've all been colorized, mostly from electron micrographs, and they're quite beautiful and informative at the same time.
Where did you find that, Dickson?
The Art in Science. I just was randomly searching for something interesting besides yo-yos in space, and since I knew that was going to be taken, I decided to get this one instead.
All right.
Skulls with walnut brains. Love it.
There you go.
Yeah, that's good.
Thank you, Dickson.
Oh, you're welcome.
My pick is not really anything to do with science, but since we encourage scientists to talk to the public about what they do, this is a website that can get you started in podcasting. Would you like to do a podcast, Dickson?
I don't know.
So this is the Podcaster's Studio, and the author is Ray Ortega, who is also an employee of the American Society for Microbiology.
Gee.
Ray came with us to Boston last week. He's a videographer and a editor and an audio editor and many other things. But he does this show where he teaches you how to podcast and goes into it pretty deeply. So if you go back to episode 1, you can learn how to do it.
Cool.
Great.
Cool.
So, Vince, how did you learn how to do it?
I'm a self-teacher.
You do
I
it
taught
very
myself.
well.
Ray didn't start, and I didn't discover him until long after I had been doing this. But I do listen to his podcast because he comes up with new stuff, because he follows the field and all the tech behind it, and I like to keep up with that. But yeah, originally I just went online and searched for how to do this stuff, and I learned. Because, Dickson, we're scientists. We can do anything.
Is that what it is?
That's the way I feel. People often ask me that.
Why is Kathy laughing?
People often say to me, "How did you learn?" I say, "We're a scientist. You can teach yourself anything as a scientist."
You
That's
can.
the point.
Do you think? I mean, I don't
No,
want
I don't
to ask.
think.
I don't want to ask our listeners to answer this question. But just between the four of us, do you think our podcasts have been getting better and better as time goes on, or have they remained the same high quality as they were when we did TWiV number 1? Because I love the way you finish the episodes and decorate them and give more information and put the art of science in each of these podcasts, Vince. You do a lot of hard work after the show is finished.
I think they have been improving, and that's why our listenership continues to increase.
Yeah.
We do cool things like go to a BSL4. We have guests, and
Yeah.
The best thing is we have lots of material to work with.
You do. You do.
There's never a shortage, right? In fact, there's too much stuff. We have a couple of listener picks. One is from Wink. What a great name, Wink. He's an MD in infectious diseases. I noticed that you sometimes do on-air internet searches and end up with commercial results. Switch to duckduckgo.com to regain your integrity. It is also more efficient. The URL ddg.gg gets you right there. Once you're ready to quack for something, try bang searching. Examples of bang searching, I don't even want to discuss this, would be quote, L Amazon Racaniello, unquote, or, quote, no, it's not an L, that's an exclamation
It's a bang
point.
mark. It's a bang.
Bang, CDC dengue. So an exclamation is a bang?
Yes.
Is that British?
No, it's computer.
The programming
really?
term.
Yeah.
See, I don't know anything about this, Dickson.
You do now?
Have you guys heard of DuckDuckGo.com?
I've heard of bangs, but I had not heard of DuckDuckGo.
Well, I went to it and I thought, what can I search for? And so I searched for blue jeans, and a sponsored link showed up. But
Yeah.
so
Yeah, I did a quick search for rabies, and I did a comparative search on Google for rabies, and I got similar, in honor of the day, I got similar results. Although I just noticed that if you do a Google search on rabies, it comes up with a little box to the side saying people related to rabies, and there are two. Jenna Geese, this girl who was the the first survivor of the Milwaukee Protocol, and Louis Pasteur.
Of course. Yep.
So Gina, besides being the first survivor of clinical rabies, Gina is in prestigious company on Google now.
yes.
What's this bang business? I don't get it.
It's used at the beginning of computer scripts to indicate the script is starting here. So
it's
you
basically a chain of search
it's
terms,
it's
right?
a couple of characters in Bash scripting. It's a £sign and a bang and an exclamation mark, and then the word Bash with no spaces between them, because £! should not occur in any normal text.
I see.
So it's a kind of a unique marker.
Well, I like the little duck on the front page.
Yeah.
That's
Yeah.
nice.
The duck's nice.
But, we have inertia with Google. It's hard to break.
And also, if you hit ddd.gg, you get a domain squatter.
Really?
Somebody's holding the domain. So unfortunately, Dickson, you're out of the loop on that.
No kidding.
Cool. DDD.
Yep, ddd.gg.
What is the GG thing?
I don't know what the GG top level domain is.
That's my email address, kids. You can't take it away from me.
Well, you could always buy it from them.
Oh, it's the top level domain for Guernsey.
Oh,
Guernsey.
okay, the country, yeah.
Country, yes.
Okay, yeah, that's TV is Tuvaluvia, something like that.
Tuvalu, I think.
Tuvalu.
Yeah.
TV domain. Let's see. I'm using Google, of course. Tuvalu. Yeah, which is a sinking island. So once the island sinks, we have to get rid of TWiV.tv.
Oops.
Well, no, you'll redirect at that point. A lot of people use the .tv.
It's in the Pacific Ocean. Okay.
And I'm very disappointed that cow.gg is not taken, since it's the Guernsey.
So all the four-letter domain names are, .com. They're all gone. So you could search for some of these. So that's TWiV.tv. I got 4 years ago. You probably hard to do that. All right. Thanks. Wink. Wink. Wink.
And his brother nudge nudge.
Josh writes, "Hello, TWiV doctors. My listener pick of the week suggestion is 3.091X, Introduction to Solid State Chemistry from MIT. This is the online version of the MIT freshman class 3.091, Introduction to Solid State Chemistry. It's free, no prereqs, and if you complete the course, you get a certificate of completion from MIT."
Wow.
So this is on edX. I've never looked at this, actually. edX, online courses. It's like Coursera, I guess. Okay.
Starts October 15th, so...
I should take this.
You should start an online course for virology, Vince.
Well, I do. And by the way, I got to tell you this. So I have my lectures online from this past semester. They're at iTunes University. So far, 56,000 students have subscribed. Wow. Isn't that awesome?
That's terrific.
Do you give a certificate?
I don't get a certificate. Maybe I should give them a TWiV mug, though, right?
Sure.
I can't afford
That's
it.
a lot of TWiV mugs.
I can't afford it.
No.
Tell them to use their 3D printer. We'll send them the program.
Yeah, it's a good idea. I could a 3D model of a mug, right?
Exactly.
That's a good idea. I realize this is not biochem, but I think it is still relevant for TWiV listeners. I took and completed the first online class, 6.002X, Circuits and Electronics. And I will tell you that they do not water down their classes just because they put them online.
Right.
It was MIT hard, and it was a brutal process getting through it. Some of us were saddened to learn that the excellent lecturer, Dr. Donald Sadoway, would not be the instructor. However, in a bonus for TWiV listeners, Dr. Michael Sema will be the instructor. It's a bonus for TWiV listeners because his specialty is more medical than Dr. Sadoway, specifically in nanotechnologies for medical monitoring and drug delivery. Here's a video where he runs down what he's working on.
Right.
In addition, EDX is offering PH207X, Quantitative Methods in Clinical and Public Health Research through the Harvard School of Public Health. 5 other courses are offered through a combination of MIT, Harvard, and UC Berkeley. Thank you, Josh. That's cool. Well, Coursera.com, I think it is. Coursera.com is another one of these massively online things. Yeah. So Columbia just made a deal with Coursera. So perhaps next semester, yes, here we are, Columbia University. There are 3 courses so far, and I have to get mine in here. Financial engineering, MOS transistors, and natural language processing.
Yeah, they have to put yours in there, Vince.
They don't, but I'd like them to. I'd like to have 150,000 people take it.
You would. I'm looking at my watch. I'm sorry that I have.
But we're done.
We are done, so I'm going to have to say goodbye.
Well, I'm not done yet.
I know, but I think
goodbye. Thank you, Dickson, for joining us.
Thank you.
Thank you.
Vincent.
Bye, Dickson.
Goodbye, everybody.
Bye, Dickson.
Yep, see you.
Bye. He has to go to the bathroom.
That's exactly right. Thank you for telling Kathy all about my personal life.
It's not personal. Everybody does that.
Yeah, they do. edX is a not-for-profit enterprise founding partners Harvard and MIT, and Berkeley just joined them.
Cool.
Hey, Kathy, a long time ago was me, Dickson, and Rich.
Mm-hmm.
And Dickson, I could tell at the end he had to go to the bathroom. I said, "What's the matter, Dickson?" He said, "I have to go." I said, "You want a glass of water?" He freaked out. Remember that, Rich?
Yeah.
TWiV is at TWiV.tv, iTunes, Zune Marketplace, and microbeworld.org. And do send us your questions and comments. We will eventually get to them. TWiV at TWiV.tv. Kathy Spindler is at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. It was a lot of fun.
Always good to have you. Rich Condit, University of Florida, Gainesville. Thank you, Rich.
Sure thing. Good time.
And Alan Dove is at alandove.com and also Alan Dove on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. WS is Western Samoa. but that's not sinking as far as I know. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Oh, you're back, Dickson?
I'm back.
Well, we're done.
What? I thought we were
Now
just
we're
starting.
really done.
Well, I'm certainly done, that's for sure.
Oh, good. Yes.
Dickson, you need some water?
No, thanks, Vince. I've had plenty.
He'll just take the empty glass.
Would you
just,
please all just divert your eyes elsewhere?
What
What did you end up naming the show?
This is going to be an Easter egg, Dickson.
What did you end up naming it?
Rabid about viruses.